My name is Colin Murphy, and I am a rising junior biomedical engineering major at Bucknell University. Today I will be presenting the effects of subthalamic nucleus anatomy on optimal deep brain stimulation locus in Parkinson's disease or PD patients. Deep brain stimulation, or DBS, involves the delivery of electrical impulses to targeted structures in the brain through permanently implanted electrodes and can have therapeutic effects on certain diseases such as PD. The subthalamic nucleus, or STN, is widely accepted as the standard DBS target for PD, but optimal stimulation location is still controversial. The objective of this study was to identify any anatomical predictors in the STN for patient-specific optimal stimulation location that would improve DBS targeting using only preoperative imaging. I used 87 PD patient STNs digitally segmented by my professor, along with each of their associated stimulation parameters. Anatomical features investigated included sphericity and two metrics for flatness generated with the major and minor axes of an ellipse fit to each STN point cloud in the three anatomical planes. The volume of tissue activated, or VTA, represents the amount of tissue in the brain receiving stimulation as a result of the electrode's active contact and was used to determine activation percentages in the three sets of STN anatomical halves. STNs were placed into two groups for each of the three anatomical directions based on DBS, electrode, active contact location, and anatomical features were compared between groups. The same procedure was followed with grouping based on predominant stimulation location calculated with activation percentages. No significant differences in any of the anatomical features were found between stimulation groups. All differences in anatomical features among active contact location groups were insignificant aside from two comparisons shown in Figure 5. STNs with medial active contacts tended to be more elongated in an axial projection than those with lateral active contacts, and STNs with anterior active contacts tended to be flatter than those with posterior active contacts. The same significant trends were not observed when grouping by stimulation location, implying some disconnect between active contact location and stimulation location. To investigate this, I grouped by active contact location and compared regional activation percentages. Dorsal and ventral active contacts were not significantly different in terms of dorsal activation, and anterior and posterior active contacts were not significantly different in terms of posterior activation. This means that stimulation may tend to spread more in the dorsal and posterior directions within the STN compared to others. In order to explain this finding, the next step for my work will be to compare fractional anisotropy values, or measures of directional diffusion bias, in different regions of the STN. With respect to the significant finding in Figure 5b, for a patient with a flat STN in the axial view, my findings suggest an electrode should be placed in the anterior region of the STN to achieve therapeutic stimulation. If it were implanted more posteriorly, the stimulation may not reach the anterior portion. Due to general lack of significance in the data, however, further research is necessary to definitively identify any anatomical predictors for therapeutic stimulation location.